properties of food products, mainly astringency and bitternes. And her research has given important advances on understanding the interaction between polyphenols, oral cavity, and salivary proteins and bitter taste receptors, and how these interactions could affect the bioavailability of polyphenols. Her work has been dedicated to develop methodologies and find new compounds to decrease or to mask such unpleasant taste generated by, in some cases, by polyphenols. And I have here a lot of interesting things to talk about her, but I think that we can stop here, and we gather that she's a great researcher in her field, and now she will. I give her the word to start with this seminar. So thank you, Susana, for accepting, and please, your turn.
Thank you, Pilar. Thank you, first of all, for this kind introduction, and also for this invitation to present the work that I have been developing in my research group about the new molecular approaches to understand food polyphenols, contribution for astringency and bitter taste perception. So I will start with a brief introduction on why is this research field so important, especially within the Sustainable Development Goals of ONU and also within the Horizon Europe frameworks. Then I will move to the state-of-the-art of astringency and bitterness. I will follow with the new developed approaches, highlighting the most important results and outcomes that we have gathered. And I will finish with my vision of what are the key questions and real challenges that future research should focus and answer regarding these two taste properties. So nowadays, food systems, worldwide governments and policymakers, as well as funding agencies, are looking to food innovation based on these four pillars: natural and healthy food, providing high quality nutrition and nourishment of population, being sustainable and with lower carbon footprints. To achieve all these, a more sustainable food system and a healthier and more sustainable diet, it is essential to place the consumer at the heart of food innovation. This implies that the first requirement is to understand the consumer perception, behavior, and choices regarding food. Placing the consumer in this key position leads to a very complex and dynamic net of inputs, such as pleasure, desire, health benefits, well-being, environmental concerns like climate changes, and the search for new alternative proteins. For all of these inputs, various disciplines are involved, such as health and consumer research, engineering, psychology and social sciences, and of course, food science. Within this framework, plant-based foods and derived products are once again in the spotlight because they align all the referred requirements. They are healthy, sustainable, and help to address climate neutrality. Despite consumer preferences, behavior, and choices are a very complex and dynamic subject, when the consumers are asked to rank the several food attributes, taste is consistently on the top 3 of the most important attributes. And in fact, this is particularly important for plant-based food products, because these products are innately linked to undesirable taste properties and of flavors, which can impair their consumption. Most of the surveys that are made to consumers usually ask them what is the importance of taste, but most of these surveys use taste as a similar thing regarding as flavor. But we know that flavor is not the same thing as taste. Flavor is a multimodal perception that encompasses the combination of modalities of both taste, aroma, and trigeminal sensations. Orthonasal and retronasal aroma are linked to two different routes for aroma perception, directly by smelling or indirectly by intake and aroma diffusion through oral cavity to nasal cavity. On the other hand, taste and trigeminal sensations are mainly perceived inside the oral cavity. Human subjects can differentiate 5 primary taste qualities, namely sweet, salt, umami, sour, and bitter, that are linked to food palatability. Sweet, umami, and salty allow the recognition of energy-rich food and are most commonly referred as food-induced food acceptance. On the other hand, the bitter taste has been linked to protection against intake of possibly toxic or harmful substances, and together with sour taste, usually is more linked to food rejection, especially if it's not perceived in balanced levels. Indeed, balanced levels of bitterness are in fact desired and expected in some food products. Regarding trigeminal sensations, they also have an important role on food flavor. These sensations get this designation because their perception occurs through the trigeminal nerve. This important sense is easily overlooked in comparison to taste and smell. Since the trigeminal nerves mediate tactile, thermal, and pain sensations, the distinction between chemical sense and tactile sense becomes sometimes blurred. Similar to tastes, also trigeminal sensations can be linked to positive and desirable attributes when we refer to the mint fresh sensation or the sparkling of carbonated beverages, and also the sensation of spicy and hot sensation of spicy food. But once again, of course, all these sensations are desired when they are perceived in balanced levels. On the other hand, astringency is also a trigeminal sensation, but is usually more linked or referred as an unpleasant mouthfeel, being considered most of the times as a negative attribute when perceived with high intensity. Bitter tasting compounds in food are mainly derived from plants, such as caffeine in coffee, hop bitter acids in beer, or soy products hydrolysates, but they can also occur in products from animal origin, like peptides in cheese, for instance. Together with phenolic compounds, the most important representative bitter compounds are alkaloids, terpenoids, glucosinolates, amino acids, and peptides. Bitter taste is perceived by a specific family of receptors that are called bitter taste receptors and that are expressed in type 2 taste bud cells. They have a sequence of around 300 amino acid residues, and they have a short extracellular N-terminus. Bitter taste receptors are G-protein coupled receptors and have a specific ligand binding sites in their transmembrane sections. This family of receptors responds to an enormous range of bitter tasting chemicals. Its transduction pathways is initiated by the interaction of the ligand with the bitter taste receptors that brings a conformational change in the receptor and triggers the heterotrimeric intracellular G-protein complex. When activated, G-alpha-gusdeucin, this subunit here, becomes released from the complex and activates phospholipase C2. This phospholipase further acts on other intracellular molecules, leading eventually to the release of intracellular calcium ions. On the other hand, astringency, as I referred, it is not a taste, is described as dryness, stifling, and puckering sensations inside the oral cavity. In foods, it is mainly induced by tannins, a specific family of polyphenols, but it could also be induced by alums, acids, metals, and currently also some works report this sensation linked to plant-based proteins. Astringency, by opposing to taste, it increases with repeated exposure. And presently, there are several mechanisms that try to explain astringency onset. But the current point of view of what happens during astringency onset is a dynamic interaction between the different mechanisms. For a long time, the most accepted mechanism is that astringency is due to the interaction and precipitation of salivary proteins by food tannins. But how this interaction and precipitation occurs? If we consider that salivary proteins and tannins have a fixed number of binding points, what happens is that for low tannin concentration, different tannin molecules could bind to salivary proteins. But these tannins could also act as multidentate ligands, binding different molecules of proteins, or even linking at different regions of the same protein. While we increase tannin concentration, this can lead to an increase of this phenomena that could result in extensive, large, and insoluble complexes. These colloidal and insoluble complexes have sequestered salivary proteins that are free in saliva, for instance, but they can also interact with salivary proteins that occur in the mucosal pellicle, this region over here. This pellicle is formed by cross-linking between proteins that are anchored or adsorbed on the oral epithelium, such as mucin proteins, and salivary proteins that are free in saliva. So, all these interactions lead to the disruption of these fluids and consequently to the loss of lubrication in oral cavity. Tannins have also the ability to bind to the oral cell epithelium directly, and another hypothesis is that tannins are able to activate oral mechanoreceptors. These receptors are buried inside the oral epithelium and are linked to the perception of touch and tactile stimuli. This hypothesis is based on a recent work that showed that astringency is perceived by activation of the trigeminal ganglion that occurs in this part of our head. However, which mechanoreceptors of the oral cavity are activated, how this activation occurs, if it is by the phenolic compound itself or by the aggregates with salivary proteins, even if the other non-phenolic astringent molecules are also sensed by this mechanism, is still unknown. So most studies on these two taste properties are performed by sensory analysis. These assays are a valuable tool, but have also important limitations and drawbacks. For instance, it is difficult and expensive to maintain a well-trained panel, and sometimes an ambiguous description between bitterness and astringency occurs. Also, these taste properties are usually unpleasant, especially when they are perceived with high intensity, and so sensory panelists can easily become fatigued. In order to overcome these limitations, different in vitro approaches have been used by our research group to study the molecular interactions that are supposed to be at the origin of astringency and bitterness. Among the most common ones used in our lab, we have the fluorescence quenching, nephelometry, dynamic light scattering, HPLC, saturation transfer difference, NMR. We also perform computational studies, and more recently we have developed cell-based assays. So, using these techniques, our research on astringency is usually divided in three main areas. We studied interaction with human saliva and or with specific and different families of salivary proteins. As I said, more recently we have established a new research area on cell-based assays. And finally, we use these established models to screen new methods or new compounds able to modulate astringency. Using this first approach, we have been able to study the interaction of human saliva with pure phenolic compounds, mixture of compounds, and also even with real food matrices. So the first work was developed 10 years ago during my PhD. At that time, we start by isolating human saliva and characterizing the human salivary profile of the main protein families, mostly related to astringency sensation, including, of course, the proline-rich proteins. At that time, this was done by a proteomic approach coupling liquid chromatography to mass spectrometry and identifying exactly the main peptides that were eluted at each time of our chromatogram. This showed us that the typical HPLC profile is roughly divided into these regions. First, we have basic proline-rich proteins that are being eluted, followed by glycosylated PRPs, acidic PRPs, then staturine and PB peptide, and finally cystatins. This first characterization opened up a huge range of possibilities for the study of the interaction with pure compounds, mixture of compounds, and food matrices that were able to precipitate salivary proteins. Moreover, this approach also made possible to isolate the different families of salivary proteins by semi-preparative HPLC to perform studies with the individual families. So, after the characterization of the human saliva profile, our first experiments were based on studying this interaction with increasing concentrations of a grapeseed fraction. So what we did was that we follow the saliva profile before this black line and after the interaction with grapeseed fraction. And we determined the percentage of decrease of the area of each family in comparison to the control saliva. We observed that the different families of salivary proteins had a different interaction with the phenolics from grapeseed fraction. From all the studied salivary proteins, acidic PRPs and staturine and PB peptide were the ones mostly precipitated by the phenolics. It was a surprise that basic PRPs were only precipitated for the highest concentration of phenolics. And this was really interesting because at that time, basic PRPs were commonly referred as the ones with the highest precipitation toward tannins. So when we compared our results with those other works, we observed that the main difference was that those works were made with purified proteins. So this work highlighted for the first time that the proteins could have a different reactivity when they occur in a mixture rather than when they occur as individual proteins. Then we applied these developed methods to study this phenomena in vivo. So to do that, we asked volunteers to collect saliva before and after intake of a grapeseed extract. We did this intake during 5 different times with intervals of 3 minutes. And we analyzed the saliva profile during this intake and also after the last sip of this solution. So this approach allowed to study from one side if the changes previously observed in vitro were similar to what happened in vivo, and also to investigate how the different families of salivary proteins may be affected along the continued astringent stimuli. We also followed what happened to our salivary profile after this astringent stimuli stopped. And why is also this important? It is important because we want to compare with the previous results in vitro, but it is also important because in literature it is described that astringency is supposed to increase upon successive sips of red wine, for instance. So we want to understand how this occurs. So here we have the results obtained for these experiments. The black bars are the control area of each family of salivary proteins in control saliva, and these 3 bars represent the CYP1, CYP3, and CYP5. So continuous exposure to our astringent stimuli. The last bar represents the 5 minutes after we stop the astringent stimulants. It was observed that repeated sips of the phenolic solution practically depleted almost all acidic PRPs and staturine and PB peptide, and also significantly reduced the amount of glycosylated PRPs. On the other hand, basic PRPs were practically not affected. So these results were in fact similar to the ones observed in vitro. When we follow the salivary profile changes upon after we stop the last sip, two results were really interesting. First, the most precipitated salivary proteins start to recover. This was more evident for acidic PRPs that start to recover after 5 minutes of stopping the exposure. And this could mean that acidic PRPs are being stimulated and released during astringent stimuli. For staturine, this recovery was only evident after 30 to 60 minutes of recovery. So this means that this protein is totally precipitated, but is not being secreted during the intake of astringent stimuli. But the other most striking result was that after the last sip, basic PRPs started to be depleted. So this raised the hypothesis that different families of salivary proteins could actually be involved in different stages of the astringency perception. In addition to grapeseed phenolics, this approach has been more recently used to study the interaction of salivary proteins with other phenolic matrices. One recent work was focused on the interaction of saliva with two Italian red wines in order to hide their brands. These wines were named M and P wines, and they were reported by wine producers as having a defect related to excessive astringency. It was so excessive that tasters were not able to discriminate the two wines. So, in addition to the classical methods commonly used to characterize red wine, the company also wanted us to apply our approach to see if we were able to discriminate between the two wines and identify why the two wines were so astringent. So we performed interaction of the wines at different saliva-wine ratios, and we observed that at a low ratio, we found that staturine, PB peptide, and acidic PRPs were the most precipitated proteins. Studying different ratios allowed us to determine that M wine induced more precipitation than MP wine, because this difference was only observable for low ratios. At high ratios, they become so saturated that all the proteins were precipitated. And probably this was the reason why tasters were not able to discriminate these two wines. Linking these results with the quantification of the specific phenolic compounds allowed us to point that the higher concentration of caphteric, caffeic, and coumaric acids could be the main responsible ones to induce a higher salivary protein precipitation and a pucker astringency sensation. So, this developed method regarding doing experiments with human saliva seems to be a method that is easy, fast, and reliable to be implemented as a quality control for wine producers, and could be implemented easily in any lab or wine facility. More recently, we have also been focused on studying how inter-individual differences on the salivary protein profile can affect interaction with polyphenols. It is known that several inter-individual factors affect astringency perception, such as flow rate, salivary protein composition, among others. So we want to deepen if different contents in the previously referred families of salivary proteins, mostly related to astringency, can impact the interaction with polyphenols. We start these studies with 20 subjects. We characterize the salivary protein profile of these subjects, and then we choose 5 subjects for which the proportion of these families was higher in comparison to the other families. So, for instance, this subject was had a higher proportion in basic proline-rich proteins, this one in glycosylated proline-rich proteins, and so on. Then we used these individual salivas to study the interaction with a mixture of prostaglandin dimers. What it was interesting to observe was that regardless of the prominent family of salivary proteins and of the total salivary protein content, the interaction followed a similar behavior. For instance, we saw that for all subjects, independent of the most significant salivary protein, staturin and PB peptide were the proteins more depleted, along with acidic PRP. So for all subjects, the behavior was similar. None of them also show depletion of glycosylated PRPs and or cystatins. So this suggests that the specificity of salivary proteins toward prostaglandins may play a more important role upon interaction with prostaglandins when compared to the total salivary protein family content and the prominence of the different families. Regarding this research line, our ongoing works are focused on study the effect of cork stoppers compounds migration to wine on the possible effects of wine astringency. We have also other works regarding the study of the salivary protein profile changes of sensory panels upon, for instance, coffee intake, and try to correlate the observed changes with the perceived taste properties. And we have also some other works in which we use other families of polyphenols and study how different families interact with the different families of salivary proteins. Moving on to the oral cell-based assays, we have established a model based on human oral cell lines that, in addition to the oral cells and, of course, salivary proteins, it also includes the mucosal pellicle. We know that oral cavity is a very complex microenvironment, so in addition to saliva, it has different epithelia that, besides having different functions, such as masticatory and taste functions, have different physiology and characteristics. And this is particularly important for astringency perception because these multiple and different microenvironments can contribute actively to this taste property, because we know that astringency is also perceived in non-gustatory epithelia, such as lips and buccal mucosa. So we have developed 2 different oral models to mimic at least these 2 microenvironments, tongue and buccal mucosa. For the tongue model, we have used this cell line, and for the buccal model, we have used this cell line. Using monolayers of these cell lines, the first step consists on the adsorption of mucin protein to the cell line monolayer. This mucin adsorption is monitored by Alcine blue or periodic acid shift colorations because they are specific for glycoproteins. And after this first step, the second step was the formation of the mucosal pellicle. The real mechanism underlying the formation of this pellicle is not totally known, but is known that in part is due to the cross-linking between proteins that are anchored on the oral epithelium, in this case that are mucins, and salivary proteins that are present in saliva. When we were establishing these models, we have monitored the salivary proteins that were being absorbed by each cell line to understand if there was any quality difference on the type of mucosal pellicle formed. And finally, the third step of our assay is the interaction with our targeted compounds, namely characterized mixtures of compounds or also food matrices. With this approach, in a 96 well plate, we are able to test two different target mixtures at 3 concentrations, and we are able to screen the interaction with the individual oral constituents, cells alone, salivary proteins alone, but also with 2 or 3 oral constituents. In this way, we are able to discriminate the effect of each oral constituent on the overall binding. Here, I present the results that we obtained with a green tea extract. We know that green tea is widely reported as a highly astringent beverage, so we prepared a polyphenol extract to screen with our models. First, we have identified and quantified the polyphenols present in this extract. As expected, this extract was rich in flavonols, mainly epigallocatechin gallate and epigallocatechin derivatives. And then we applied this extract at 3 different concentrations. Here, I'm presenting only the results for the highest concentration and only for the epigallocatechin gallate compound. Within all the identified polyphenols compounds, it was observed that GCG, his methylated form, and also epicatechin gallate were the compounds with the highest interaction. Regarding the effect of each oral constituent on the interaction, it was observed a similar effect for all compounds. So all compounds interacted in the same trend. Here in black, we have the initial concentration of epigallocatechin gallate in the extract, and the other bars refer to the concentration that was bound by each oral model. The interaction seemed driven by the presence of salivary proteins. And this means that all models that had salivary proteins in their composition were the ones with the highest interaction. Here, this is represented by the models in the buccal mucosal model, this one that has all the components, oral cells, mucosal pellicle, and salivary proteins, and this one that has oral cells and salivary proteins. And we see that the interaction was similar in buccal mucosal model, but as well as in tongue models. On the other hand, when we have only cells or cells with mucin, we see that there is a lower interaction. But what was also interesting on this model, on this interaction, was that there was no difference on the interaction between these 2 cell lines. So we see that cell line alone, the buccal cell line, has the same interaction, statistically speaking, than the tongue cell line. We have also applied this method to study the interaction with a red wine anthocyanin extract. First, we have identified also and quantified the anthocyanins present in this.
So now we don't see. Now we can see. Sorry. Okay, go on. Sorry.
Okay. Please stop me if anything goes wrong. Okay.
Of course, no. It was just maybe it was just me. Go on. Sorry.
Okay. So in a similar way, we start to identify the main anthocyanins present in this extract. As expected, malvidin-3 glucoside was the main anthocyanin, and then we also applied this extract at 3 different concentrations in our models. Within all the anthocyanins present in the extract, we observed that malvidin-3 glucoside was the compound with the highest interaction. And regarding the effect of the oral constituents, we observed that for all anthocyanins, the interaction was higher with cell line alone. It was also interesting that somehow the presence of salivary proteins, either the ones present in saliva and also mucin protein, restrained the interaction with cell lines. Once again, there was no difference between these two cell lines, so we observed that the interaction with the buccal mucosa and tongue cell line was quite similar. The results with these both extracts and the fact that there was no difference on the binding between tongue and buccal cell line raised the hypothesis that the differences on astringency perception may arise from the ultrastructure of the tongue. This is differences on the other buried epithelium structures, like the mechanoreceptors. And it is also important to highlight that these studies, apart from being very recent, they are a turning point on astringency studies, because our data allowed us to study the contribution, to monitor and study the contribution of each interaction, the effect of each oral constituent and each polyphenol interaction when these interactions occur all together, more similar to food matrices and also more similar to an actual food intake. In fact, this approach has been also applied to screen commercial coffees from different brands that were also analyzed by sensory analysis. These results are now being under evaluation because there is a lot of data. But just a quick overview to show that the different coffees showed a different interaction with our models. And more important, we know that there was a strong correlation between the total bounded phenolic compounds with the perceived astringency. So these models have a huge potential to highlight from one side the contribution of each oral constituent in astringency, but also to identify which compounds within a food matrix are interacting inside the oral cavity. Ongoing works regarding this research area. We are now with a project that studies the effect of wine oxidation that intends to simulate wine aging on the interaction with salivary proteins and also with these cell-based models. And we have also some other research with other food matrices to apply them with our models and correlate this with sensory assays. And finally, I'm not going to present any data regarding the modulation of astringency, but we have also been using these two models to identify polysaccharides that are naturally present in food or that can be isolated from food byproducts or other byproducts that have the ability to modulate all these interactions, the interactions with salivary proteins and also the interactions inside the oral cavity with oral cells and mucosal pellicle. Regarding bitter taste, we have been working also in three main areas. We have been using a cell-based assay to express bitter taste receptors and screen their activation by different families of phenolic compounds. We have also started to study the effect of salivary proteins on this activation. And finally, we have been also trying to develop a specific biosensor possible to be used directly in beverages industry to assess this taste. The cell-based models that we have been using so far is based on cloning the bitter taste receptors into plasmids, which are then expressed in egg cells. Then these cells are placed in presence of the compounds to be tested, and after activation of these receptors, this induces a signal transduction pathway via G protein that culminates with an increase in intracellular calcium. This increase is perceived by a specific probe that becomes fluorescent in presence of calcium. So At the end, beta-taste receptors activation is measured only by fluorescence signals. This work has been developed in collaboration with the German Institute, but currently we are implementing this approach in our lab. We are doing some changes in this method to get a more realistic model of oral cavity, because most of the researchers that use this approach, they are based in egg cells, which are quite different from oral cavity cells. And so we are trying to mix this model with the cells used in an astringency model to get a more realistic approach of this taste property. Using this in vitro assay, a library of polyphenol compounds from different families, condensed tannins, hydrolyzable tannins, anthocyanins, and ethyl esters, was gathered to study the activation of these taste receptors. We found that one or more beta-taste receptors were identified as responsive for these individual compounds. Overall, 7 different receptors were activated by 12 polyphenol compounds. And it was interesting because different compounds activate the same receptor, and also the same compound activate different receptors. The most interesting case was for EGCG, that activated 5 different receptors. But what is really also interesting in this table is that we can see an overlapping or similar activations within a polyphenol family. For instance, condensed tannins, almost all members activate receptor 5. Activate receptor 5, while almost all hydrolyzable tannins activate receptor 7. So there could be here some specificity of polyphenols family to beta-taste receptors. After determining the responsive receptors, it was established the dose response curves for each agonist receptor pair and determined the half maximum effective concentrations. From these values, we can see a wide range between micromolar to millimolar range. We observed that among the library studied, granidine was the compound with the lowest EC50, while epicatechin was the one with the higher EC50. It was also interesting that if we focus on one compound that activated several receptors, we see that the EC50 are quite similar between the different beta-taste receptors. For instance, epicatechin activated all these receptors in the millimolar range, while EGCG activated all of them in the micromolar range. Finally, to finish the results, for some of the polyphenols that have been already previously known to interact with salivary proteins, such as epicatechin, PGG, and malvidin-3 glucoside, we asked if this interaction with salivary proteins could affect the activation of beta-taste receptors. So we choose to study the effect of salivary proteins at 2 polyphenol concentrations, one near DC50 and the other one at the maximum of the dose response curve. And for all cases, it was observed that the presence of salivary proteins, the data here in blank bars, we showed that for all cases, the presence of salivary proteins reduced the activation of beta-taste receptors, and this was in fact independently of the polyphenol concentration. So, to summing up all these results and all the information that our work has been gathering, I want to highlight that these cell-based models on astringency studies are a turning point, because our data allows to study and to know the contribution of each oral constituent on the overall interaction, plus know which compounds inside a mixture are really interacting with our models. We now have been gathering some results that showed us that individual compounds could have different behaviors, but The most important results show that it seems to occur a relationship between the polyphenol family and specificity toward oral constituents, oral cells, and also different families of salivary proteins. So, since different families of phenolic compounds have also been reported to have different astringency mouthfeels, one hypothesis that is on the top of the table is that different interactions can be related to different astringent mouthfeels. Joining this approach to sensory analysis has a huge potential to highlight the contribution of each oral constituent to astringency mouthfeels, and even to deepening the mechanisms underlying astringency perception. Regarding bitterness, we have now the point of view that the interaction with salivary proteins affects bitter taste perception, but we need more realistic models, and we are now working on this topic with a PhD student, actually. So this is a summary of the research that I will be pursuing in the next years. I intend to do all of these areas of research, considering the approach, considering always the human approach, linking all these molecular assays to sensory analysis, looking from a perspective regarding what happens in cells. So we are also developing even more realistic models, trying to include also mechanoreceptors on our models, and more representative physiological models. And we are also always interested in understanding the interactions at the molecule level. So we are also developing work in characterizing the epitopes of binding regarding astringency and also regarding bitter taste receptors. And of course, that all of this only has interesting when we want to focus in modulating these properties toward consumer acceptance. I will finish presenting our group. So the heads of our group are Professor Vitor and Professor Nuno, and the team that is directly related to astringency and bitterness research is this postdoctoral research, Dr. Elsa Brandão, and also these two PhD students, and one master fellowship that hopefully will become a new PhD student. So, and I will finish. Thank you all for your attention, and I am available for any questions that you want.
Oh, thank you very much, Susana. It was a great presentation. I'm very happy to hear all that you have told us, and I see that we have several groups in our institute that will eager to collaborate with you and your team. So I guess that we have some questions over there among assistants. Please raise your hand if you have any question, or just open your microphone. Okay, go ahead, Juan.
Okay, so yeah, very nice presentation, Susana. So I have several questions, but maybe I can start with the last part. So so this effect that you see of modulation by the salivary proteins of the bitter taste. So do you think this is happening at the? So I mean, thinking in the mechanism. This is happening at the receptor level, so at the extracellular reception, or is an intracellular signal pathway that somehow is compensating? So both signals are somehow competing. Do you have any idea of?
Not yet. We are trying to understand that part. If it is a competition, or if it is a signal change. Yes, these are only preliminary data because we had to adjust all the cell assay, because we could not actually do this with the whole saliva. So cells are extremely sensitive, and so we had to make artificial saliva with human salivary proteins. But we are now trying to understand what is really happening. This is the research project of one PhD student.
So, and something that is related to this, so are you able to also to make mutations of these receptors in your cell system, or for now you are only analyzing the wild type?
For now, I'm only analyzing wild type. I have some already some haplotypes for. I'm working in this regarding the effect of salivary proteins. We are working with the 3 receptors that were mostly activated by polyphenols compounds, and so I have also now haplotypes for some of these receptors, in particular for receptor 5. But for now, we are just doing this. Maybe in the future we can try to do some mutations.
Yeah, I think that would be very interesting if we have models, no, that
yes.
we can try to model that in the computer. It would be very interesting, no, to see the effects. So if I can make another question. So you showed the 7 receptors, so the results for 7 receptors, but I didn't understand if you checked also other receptors, or is so what happens with the rest of the receptors?
We checked the 25 receptors, all of them, but only 7 were activated. But when we go to the dose response curve, sometimes we are not able to get a dose response curve for all the activations due to unspecific signals. So that's a major problem, and that is the main reason why I am trying to change this this assay that I did in Germany, because those cells are really sensitive to unspecific signals, and so this limits their application to some polyphenols. And so I'm trying to develop now the same system, but using other cells that are more resistant, to try to study other polyphenols that it was not possible with the other approach.
Okay, I understand. So it might happen that so the rest of the rest of the receptors that you didn't find any signal, so maybe you try other polyphenols.
Yes.
Yes. You could. Okay. Yeah. I mean, I think I stop by now because just in case anybody asks.
Okay.
Thank you.
Thank you for your questions.
Any question more over there among people, the audience? Okay, if no one asks, I have some questions for you, Susana. So as I said, congratulations for your presentation and your very hard work. And I'm surprised about all the back things you have done. And I have some questions related to, because here you already state that anthocyanins like malvidin, the most abundant, is involved in both bitterness and astringency. So okay, so it's great because we have been working on this part, and we have with another approach, and we have also seen that they are involved in bitterness and astringency. When we speak white wines with the anthocyanin fraction, we see that there's an activation or an increase in some mouthfeel perceptions, not related to dryness and other sensations like dusty or sticky. So this is very in line with your in vitro results that we are confirming from the other way around, from the product and what we are sensing, and you are working more at the molecular level. So I'm happy to hearing, and we should transmit and say that the winemakers, that malvidin or anthocyanins are involved in such sensations when they are looking for getting more and more color in their wines. And I would say, okay, but be careful, be careful. We are not yet sure which compounds will, which anthocyanins will modulate each sensation of, and if you are increasing your intensity in terms of color, but what's happening with bitternes and astringency. So this is a
that is really, really interesting to hear. This complementary with sensory analysis is what I'm looking for now, because now we can have the details on which compounds are really interacting, but we do not do sensory analysis. And I think that there is really a complementary on this type of research. Yes.
And also we are seeing what you are seeing, but your hypothesis that different families of polyphenols are driving different mouthfeel sensations. We are seeing the different mouthfeel sensations, and then we are seeing that tannins are driving different sensations than anthocyanins, and it's very well in line. And we should maybe look more inside each kind of anthocyanin and each kind of tannins. And to this regard, I have seen that you are working on the astringency in oxidation.
Yes, that is a collaboration. Maybe you know the researcher Alexandra Rinaldi.
Yes, in Italy they are also with mouthfeel sensations working.
Yes, this is a project that we are doing with her. She's doing the oxidation of wines and monitors all the typical analysis of wines, and we do the cell-based assays with her wines. So,
and which are your hypotheses?
Sorry.
Which are your hypotheses concerning oxidation? Do you think that oxidation increases astringency perception, or?
So far, we are still doing that project, so we do not have any final results to discuss on what is happening. We see some shift of interaction with salivary proteins. There seems to be different families that are precipitated by oxidized wines. So I think that probably they are less astringent, but we are still looking to the results.
Okay, there I have also a remark there, because in oxidation there's an increase number of volatile aldehydes. That these aldehydes have been we have been working on the green character of wines, and we have seen that these aldehydes are driving trigeminal sensations that maybe interact with astringency perception. So maybe if you are working with wines, I would suggest to make the sensory work, make it with nose clips, because their aroma compounds are maybe driving some trigeminal sensation and also interacting with your astringency perception and bitterness. So it's nice hearing this work with oxidation, and I will be there for hearing your results. And I have no more questions. If there's someone who wants to add anything, please raise your hand. No? So again, thank you, Susana, for your the great effort you did, and please go on working as you are doing, because it's a very brilliant and great work. And please, we are very open to have collaborations, our institute with you, and please feel as if it were your house in case you want anything. So
thank you so much for this invitation and for your kind words, and hopefully we will collaborate. I think so. We have a lot of things in common, so I think it will be really, really a pleasure.
Okay, so thank you, and we close this session. And as you see, the comments from Javier Ibañez, from Hugo, and they are very happy with your.
Thank you all.
So,
thank you.
Thank you. Keep
so
safe. Keep safe, and
you
see
too.
you soon, hopefully.
Yes. Thank you. Bye
Bye.
bye.
Thanks very much. Bye.